Okay, hello everyone, and welcome to the first in a series of 4 CCDC webinars on the subject of solid form informatics. Before I introduce our speaker for today, I just have a couple of technical announcements to make. First of all, can I please just ask that you all save any questions until the end of the webinar. The phone lines will be muted in just a moment, and that will continue for the duration of the presentation. And at the end, we'll open up the phone lines and invite you to ask some questions. And secondly, we are recording this webinar, and we'll make that recording available from the CCDC website soon after the event finishes. Okay, so our speaker today is Colin Groom. Colin is the Executive Director of CCDC, and prior to that he had a career in the pharmaceutical industry, first with Pfizer and then with Celtech, which later became part of UCB. And given Colin's unique background, he's going to be able to give us some insights from both the industrial perspective and also from the CCDC on the role and the potential impact of solid form informatics. So I'll pass you straight now over to Colin so he can begin.
Thank you, Gary, and welcome to the first webinar in this series. Now, I intend to introduce the research that's been performed at the CCDC over several years in the area of solid form informatics. This will then lead on to an introduction of some of the tools which have been developed and made available through the solid form module of the program Mercury. More specifically, I'll discuss how we can extract knowledge from the vast amount of data in the Cambridge Structural Database and apply that knowledge to allow material scientists to make better decisions during the development of new compounds. Although my examples will be drawn primarily from the pharmaceutical sector, the techniques I'll introduce are equally applicable to agrochemical development and indeed the development of solid forms of any chemical compound. But I'd like to start with this quotation from Professor Steve Byrne from the Department of Industrial and Physical Pharmacy at the University of Purdue. He says that not only is he says that the solid state chemistry is tough to get right, but I'd like to add to this that it's also vitally important to get it right, and we'll see why shortly. Now, at the point in the drug discovery process where solid form development begins in earnest, an organization's made a real commitment to a compound. In the case of drug discovery, we can try many compounds, selecting the best for onwards development, and just discarding those we don't find suitable. That's not the case in drug development, where typically there's one development candidate for a project which needs to be formulated into a drug. The challenges presented by that compound need to be addressed head-on. You can't simply choose an alternative. It's also the stage of drug discovery where time pressures really begin to be felt. It's the point where time does start to mean money. It's also a process which must be gotten right, with serious consequences if this isn't so. In other words, the development of a compound represents a risk. Now, let's look at risk. Every industry has its own risks. Let's just look at some of them. Power generation is one. It's difficult to mitigate against the impact of a catastrophic failure. But the likelihood of this happening can be addressed and is by power companies that take it incredibly seriously. The same's true of surgery. We'd like our heart surgeons, for example, to take informed decisions on solid data to determine the best course of action. Particularly, we want them to reduce risk. Now, when we fly, we expect our air traffic controllers to minimize the likelihood of collision, because that collision can have such a significant impact, literally. And of course, the same is true for rail travel. Indeed, we've come to expect risks to be not just minimized, but to be eliminated. What about chemistry? Well, of course, there's obvious safety risks of practicing chemistry, but the products that we produce also present risk. Now, one of these risks is the tendency of organic molecules towards polymorphism, and that's what the bulk of this webinar will be about. Sure, you're aware of the concepts of polymorphism, but for the purpose of today's webinar, this is what I mean when I refer to polymorphism. The particular point I'd like to highlight here is that polymorphism is the norm. We should expect organic molecules to exhibit polymorphic behavior. We can't reduce the likelihood of polymorphism, but what we can do is understand which polymorphic forms are possible and select the most appropriate. This slide illustrates why an understanding of polymorphism is so important by listing some of the properties of a substance which can be influenced by the polymorphic form. Many of these are crucial for the performance of a drug substance, for example, the stability, the solubility, the dissolution rate, and the like. Now, of course, those who regulate the industry on our behalf are keen that organisations properly recognise and address polymorphism in drug substances. What we hope is that combining the experimental studies that people do with the type of analysis I described today will allow organisations to demonstrate that they have a thorough understanding of the polymorphic landscape of their products. So let's look at some real examples. The first is from the biopharmaceutical company UCB, and I believe we have some UCB colleagues on the line who will undoubtedly be very familiar with this. So in 2006, UCB acquired Germany's Schwarz Pharma for $5.6 billion. Now, one of the chemical assets of Schwarz was the compound reticotine. This was brought to market as Nupro, which was a treatment for Parkinson's disease, and also showed promise for restless leg syndrome. Now, as we can see from the extract of this patent, it was reported that reticotine didn't show any polymorphic behavior. Reticotine was formulated to be applied as a skin patch, which had tremendous benefit to patients, giving 24-hour exposure to a compound which actually has a relatively short half-life. Let me show you a video. It's just from YouTube, and it's run by the FDA, describing what happened next. Now, don't worry if the video doesn't work too well through your WebEx. It is available on YouTube, and I'll show you some pictures afterwards. So let's just take a look at this.
Schwarz Pharma recalled Nupro Transdermal Patches at the end of April 2008. These patches are used to treat early-stage Parkinson's disease.
The problem is that reticotine, the active ingredient, may crystallize in the patch. If that happens, less drug is available to be absorbed through the skin, and so the efficacy of the products may vary. The crystallized drug may cause a patch to have snowflake-like patterns. The more snowflakes, the greater the chance that the patch won't work properly.
Because of this, new patients should not be started on Nupro. Patients already using the patch should be slowly titrated off it and started on an alternative treatment as needed. Patients should not abruptly discontinue treatment with Nupro. The down titration should proceed according to guidelines in the labeling, using patches that are unaffected or minimally affected by the crystallization. Patients should be told to look at their patches and use only those that have this amount of crystals or less. For more information, you can contact the company at 1-800-477-7877, option 9.
So here again are these troublesome little snowflake crystals growing within the transdermal patches of Nupro. Now, of course, this had an effect on the stock price of the company, which you can see by the red line here. But more importantly, it had an effect on patients. This is a composite screenshot from the web pages of a superb blog maintained by Bidmona, who happens to be a Parkinson's sufferer. So just read what the Nupro withdrawal meant to her. She says, "There's no way I can go back to the oral requip. Those were miserable days. Dealing with the tremors and balance issues are bad enough. Vomiting all day long really shouldn't be an option for a Parkinson's patient. This had really serious consequences on people." Sadly, it's not the only case of this happening. A decade earlier, Abbott had to withdraw their HIV treatment, ritonavir, from the market. In this case, a new polymorph appeared in the manufacturing process, a polymorph that dominated from then on. Again, we have real experts on this on the line from Abbott. I think, once again, the impact's best appreciated from a patient's perspective. In this case, I've selected a few excerpts from the blog of Jules Levin, who's the Executive Director of the National AIDS Treatment Advocacy Project. Now, he described a meeting where Abbott presented the challenges they were facing with the manufacture of ritonavir. Once again, the key issue related to the solubility of a new polymorph. And in this case, despite really heroic efforts, Abbott weren't able to go back to the successful manufacturer of what was then known to be a metastable form. Jules continues, and what's interesting is the comment from Steve Byrne, highlighting that this appearance of alternative polymorphs isn't uncommon. doesn't always have an impact on manufacturing or on patients, but not knowing of the likely existence of more stable polymorphs can have a major impact on intellectual property. Now, all of these cases inspired CCDC to see whether the hundreds of thousands of structures in the CSD would enable us to develop predictive tools for polymorphism. Here's Eugene again, who we came across in the ritonavir case, making a wonderful comment where he likens polymorph prediction to hurricane prediction. Now, of course, as he says, we can't prevent hurricanes. We can't prevent polymorphism, but we can begin to understand the likelihood of them happening and the associated impact. This next slide shows that in the years since Eugene made this comment, the prediction of the track of hurricanes has improved to a point where it allows us to make preparations, be they to evacuate, organise emergency responses, or to stock the shelves in one of our stores. Now, we'd like to apply the same type of approaches to polymorphism. But in order to perform predictions of this type, we need data, lots of data. We'd like to use this data not only to determine risk, but also to allow us to exercise more control over what we do and take appropriate decisions as we develop compounds. What we're going to be talking about is nicely captured in this book from Microsoft. It's a collection of essays that expands on the vision of the computer scientist Jim Gray for what he called a fourth paradigm of discovery. This is based on data-intensive science. And actually, there's some lovely examples in this book about how we're now in a position to develop new tools and understanding from the data that we actually already have. The data, of course, that we care about is in the CSD. Now, the growth of the CSD continues apace, as shown in the histogram on the left, and we have great coverage of pharmaceutical compounds, as shown in the pie chart on the right. Eating this pie would have quite a profound effect on you. The contents of that pie are shown on the next slide. What I've done is taken this lovely poster describing the top 200 selling pharmaceutical compounds and coloured those for which we have a structure in the CSD in pink. Those in white represent structures yet to be deposited with us, and those in grey are treatments which are small molecules. So this demonstrates that not only do we have lots of data, it's data that's relevant to the problem in hand. In this case, it's that of the polymorphism of organic compounds, and most particularly the polymorphism of pharmaceutical compounds. Now, that's what solid form informatics is all about. It's about using statistical tools to turn the data we have on over 40 million atom-atom interactions in the CSD into actionable knowledge that we can use to guide our science. So one of the first examples from the CCDC was to look at hydrogen bonding patterns in drugs and develop a tool to understand the propensity of an interaction between specific groups in a specific compound. This work represented two key developments. The first was the ability to create subsets of data from the CSD that were relevant to our specific compounds. And that's only possible now the CSD has grown to such a big size. But it also shows the ability to turn the knowledge we can extract from the CSD into numerical algorithms that we can use to develop software tools. We've joined those software tools together and created what we call the CSD Solid Form Suite. So, how can I help? Well, let's look at some examples. Let's return to ritonavir and see how these tools might have helped there. Our hydrogen bond propensity tool identifies all of the possible hydrogen bonding groups in a molecule. They're coloured in grey here. And it then identifies all of the structures in the CSD which contain relevant information. In the case of ritonavir, there are about 1,000 structures that contain at least two of these groups. We can automatically look at these structures and we can calculate the propensity for any particular hydrogen bond to form when we have this specific repertoire of hydrogen bonding functionality. The key development here is to look at the hydrogen bond propensities in the context of this restricted array of groups. So let's look at those propensities. The column here labelled pi effectively gives you the probability of every individual hydrogen bond forming. So we can see that the most likely interaction between these types of functionalities is an amide carbonate hydrogen bond. Interactions with a low pi value are seldom observed, when these groups have other possible interactions that they could form. So we see that the original form of ritonavir, form 1, has two really unusual hydrogen bonds between the urido groups and the hydroxyl group, and also between the urido groups together and between the hydroxyl group of one molecule and the thiazole group of another molecule. These are really unlikely. So with the knowledge of these values, it should no longer be a surprise that more stable forms are possible. In this case, we see this troublesome form 2, which doesn't form these unlikely interactions. In fact, the hydroxyl group is able to interact with an amide, a much more usual situation. In fact, there are now 5 forms known of ritonavir. Let's look again at reticotine. Here it isn't the interactions that give us a clue to potential polymorphism, but unusual geometry and disorder. The crystal structure of the original form of this drug revealed a disordered thiophene ring that adopted a somewhat unusual conformation. So the green bar chart you see is the output from the CSD system program Mogul, where the torsion angles of all of the crystal structures containing an alkyl-linked thiazole have been compared to our target structure, which is shown by the red bar, the red line. The form observed to crystallize in the transdermal patches has a different, more usual conformation shown by the red arrow. And it may well be that unusual conformations are indicative that a more stable polymorph without such geometric compromises may well be possible. So, in those 2 examples of ritonavir and reticotine, we've looked at suboptimal hydrogen bonding and conformations that have been unusual, and we've suggested that they may be indicators that a more stable polymorph could exist. Let's look at another example. What we've done here is to look at the crystal structure of sulfothiazole. Here we've automatically calculated the positions we would expect to see various groups of neighbouring molecules of sulfothiazole in the solid form. What we can clearly see is that one would expect the sulfonamide to be positioned in the red blob that we see towards the top right next to an amine group. Now if we expand the crystal lattice, what we see is that this group isn't in that position, and it gives us a clear signal that other, more stable polymorphs might be attainable where a more optimal packing is observed. Another property that can be assessed through the application of structural informatics is the quality of the hydrogen bonding. Poor hydrogen bonding definitely raises a flag that there may be a more stable form available. What I've done here is plotted the proportion of hydrogen bond functionality that's satisfied in one polymorph of piroxicam, which we refer to as participation, against the summed likelihood of each hydrogen bond interaction, which we call the propensity. And this particular crystal form is represented by the pink dot. Now all the grey dots are other possible combinations of hydrogen bonding groups, each given participation and propensity values. Now we predict that lattices with a more negative participation value and a higher propensity value, i.e. those in the bottom right of this plot, are likely to be the most stable. Good hydrogen bonding with the expected participation from functional groups. Now this polymorph that we see in pink is clearly not a good prospect. There are other structures which have much better functional group combinations, and I guess one would be advised to seek other polymorphs if one's drug candidate was sitting in this area on this chart. The key message here is that the placement of the polymorph on this chart indicates it's not likely to be stable. Indeed, there is another alternative polymorph for piroxicam, shown again on this diagram by the pink dot. Now this one has the most optimal hydrogen bonding possible and is a more stable polymorph. It would represent a lower risk prospect for onward development. It's much more likely to be stable. So this type of quick visualization can be invaluable. Here's another use of this hydrogen bond propensity tool. In the previous slides, we've looked at the position of the pink spot on the chart to get information about likely stability. For this example, we're looking at temozolomide. So the database analysis here shows that there's a cluster of possible structures, all with good hydrogen bonds, and all where the functional groups are participating at expected levels. This indicates that polymorphism may be an issue. There are a number of plausible outcomes. Indeed, there are at least 3 known polymorphs of temozolomide, and all of the hydrogen bonding patterns observed in those structures are found within this cluster identified. Here's a great example. It's the drug omeprazole. It's the world's current top-selling drug. The form we see in the CSD, and the form that's indeed marketed, has optimal hydrogen bonding. all of the possible forms as suboptimal, represented the best solid form for onward development. We can see the separation of the observed form, again in pink, from the other structures. That indicates that finding another stable polymorph of omeprazole really isn't that likely. Of course, it's not all about reducing risk. This type of analysis can allow one to identify new opportunities. So let's take lamotrigine on the left here. We can calculate whether the hydrogen bonding groups in a coformer, what I've shown here is methylparaben, we can calculate whether those hydrogen bonding groups will outcompete those of lamotrigine itself. If they do, it would indicate that a co-crystal might be possible between lamotrigine and methylparaben. And indeed, that co-crystal structure does exist, and it actually has some improved properties. For example, it's more soluble and has bioavailability. So this type of analysis, using the CSD to rationalize the experimentation to pick the likely coformers, found new co-crystals and has actually resulted in patent applications. And we'll refer to this in subsequent webinars. This sort of interaction analysis can be complemented by looking at the overall molecular properties of potential coformers as well. Whilst at the CCDC, Laszlo Fabian performed an analysis of co-crystals and determined the important properties of compounds which needed to be complementary in order to form co-crystals. When this was applied to a real-life co-crystallization experiment, he found that 85% of all co-crystals could be found by performing only half of the experiments, those experiments which CSD data would suggest were the most likely to be successful. Of course, you might well say that given the great progress in crystal structure prediction described in papers such as the one at the bottom of this slide, which is involving the CCDC and the group of Sally Price at UCL in London, that we should focus our attention solely on CSP. Now, in CSP, crystal structure prediction, predicted structures are usually plotted on diagrams such as this, where the relative total energy is what really matters, but the structures are spread out by their calculated density. Those at the bottom of this plot are calculated to have the lowest energy and should therefore be the most stable solid form. Now, whilst it is possible to predict the structures of some compounds, those compounds for which there are a number of possible structures closely related in energy present an ongoing challenge, as do more complex crystal forms such as salts. Now, we're currently in a situation where this might happen. We might predict that the green structure on this plot has the lowest energy. We might conclude that that's the most stable polymorph, and we might choose to develop that form. But as we improve our methods, it's quite possible that we subsequently find that it's the red structure that is actually the lowest energy, and we wish we'd developed that. So a prudent approach might be to combine these types of calculations with the solid form informatics approaches which we've described today. As an aside to this, I would encourage you to look out for the results from the latest blind test of crystal structure prediction hosted by the CCDC, which are about to be published in ACTA Crist. So, to summarise, applying solid form informatics techniques can be of tremendous benefit in the development of novel compounds, or novel forms of existing compounds. The techniques are based on the entire published and deposited output of small molecule crystallographers. That's some 600,000 crystal structures. They not only allow us to rationalise the observed behaviour of compounds, but also to predict that behaviour in advance. Of course, we could continue to work as we have done, but that does leave us with the risk of late appearing polymorphs, and also means we may fail to spot the solid form opportunities that lie in front of us. I'll conclude with this summary of how solid form informatics can guide us to making good decisions at various stages of compound development, hopefully decreasing the uncertainty and risk that really pervades in drug development. So these tools should allow us to move from a position where we do experiments because of fear to one where we do experiments to validate our hypotheses. Now, this was just an introductory webinar. If you found it of interest or think it would be of interest to others, please do register your interest on this webpage or send us an email to this address, and we'll provide further details of how we might be able to help you, or we'll give you the opportunity to try out these tools for yourself. I'd also like you to draw your attention to our Crystal Form Consortium, which is a group of leading pharma, development and agrochem companies who've helped steer the development of lots of these tools and have had the benefit of them for a little while now. This consortium is now entering a new phase where we'll appoint a research scientist to work with our individual consortium partners on addressing their specific issues and needs, and also work on furthering developing these types of tools. Again, please email solidform@CCDC if you think you might find this interesting. So, this webinar will be followed by one from Bob Doherty, who's the Director of Material Sciences at Pfizer and Sandwich. And he's the person who really spearheaded the development of solid form informatics. In fact, it was Bob who introduced that very term. We'll then have two more technical webinars where Elmer Pidcock and Pete Wood will take us through the science and the operation of some of our tools in a little more detail. With that, I'll hand you back to Gary. Before I do so, I should perhaps thank you for taking part in this webinar, and I'd like to thank all of my colleagues at CCDC and many people who we've collaborated with, in particular our Crystal Film Consortium members and Pfizer as part of the Pfizer Institute for Pharmaceutical Material Science, who've really allowed us to develop this science.